Final speaker is Robert Henry from Queensland, who's going to talk about genome sequencing in relation to germplasm.
Thank you, and I too would like to thank the organisers, especially for accommodating the fact that I've come late from the meeting that Dave Edwards was organising in Amsterdam on agriculture and climate change. So today I'm going to talk to you about some of the systems that we're studying and really how genome sequencing really underpins and is an important approach for us in characterising germplasm and really getting value from it. I start from the technology, and I think it's really important to recognise that technology is continuing to change, and it doesn't seem to me that it really is slowing down. We keep thinking every time there's a big advance that, well, that's a great thing and we'll be stable for a while. They can't top that, but we are. And some of the things that we've been doing recently is really looking at the accuracy and comparing what you get from some of the different platforms. And I think we're learning that there actually are strengths and weaknesses in all of them. And it's great that we've actually got a range of technologies, and I think that creates an environment in which we're likely to continue to see significant advances in DNA sequencing technology with the cost continuing to come down. I'd like to define it in terms of a number of publications I've been involved with that really, I think, define how the technology has advanced this century from about 2000. And if we go back to to 2000 or the turn of the century, we were really using microsatellites and just starting to think about moving from microsatellites to SNPs. In the early noughties up until around this time here, we saw a lot of transition away from microsatellites to developing SNPs, but we really were limited in our ability to discover them. We had great tools for analysis, but discovery was limiting. But what happened at this point, of course, was we had the emergence of next generation sequencing and these big advances in sequencing technology. So from 2008 to this book in 2013, we saw a complete transition, and now, of course, nearly all of the markers that we find are being derived and discovered by next generation sequencing. But last year put out a book in which we really defined, for example, a protocol for finding all of the differences between any two individuals. And I think that the way that we do that is continuing to evolve. It's interesting to see that a year later we do it very differently, and in a year's time we probably do it very differently again. What I see is a complete convergence of genotyping and sequencing, so that really genotyping an individual will, in the not too distant future, be obtaining its genome sequence, because the ultimate genotype is that genome sequence, and we won't be satisfied normally unless we have the whole genome sequence for each individual, because it's becoming increasingly a realistic thing to do. For some of the smaller genomes, things like rice, we're just starting to apply that approach of saying that the way we'll genotype is to resequence the genome of each individual. And I think that we don't need to project very far forward to see that becoming the more common strategy for genotyping. I'd like to talk about a range of systems, including how I see some of this helping us to domesticate new crops or re-domesticate existing ones, and we've just heard some examples that might be put in that category with the chickpea work. And I think we really have the ability to with these technologies, identify the domestication genes, and we've been working on distant relatives of rice that might provide new temperate rice substitutes and other crop wild relatives, and accessing the genes that we haven't been able to access before. Effectively, what we've been able to do in the last few years is characterise most of the domestication genes such that we can look to domesticate a crop not in thousands of years, but in 1 or 2 years. For example, any of the grasses out there, if we wanted to turn them into a cereal, we really would be able to deal with shattering, grain size, a whole lot of classical domestication traits, and do all of that in essentially 1 or 2 generations. So we can take them from the wild into a domesticated form that would perform in agriculture. And so we do have the possibility, I think, particularly when we're considering the need to adjust agriculture for new climates in the future, to be able to take new species and use the knowledge we're getting from genome sequencing to domesticate new cropping options. I'd like to say a little bit about what we're doing with the major cereals. First of all, rice, and we've worked with the many species in the ICRISAT genus, but more particularly recently we've been working with the Genome species, and these are the ones that are the closest relatives of rice. They represent the gene pool because these are the species that are interfertile with rice and the ones that we can easily access for genetic improvement. And our main focus has been on characterising the emerging diverse material we're discovering in Northern Australia, a number of taxa populations that we believe are new species, and we think that there are many more species yet to be discovered in the ICRISAT, particularly those that are among the closest relatives of cultivated rice. And what we're finding is not small populations. We're seeing that there are very large populations in the wild of rice that is undomesticated. And the unique feature we have found in these Australian populations is that they are genuinely undomesticated. Much of the wild rice that we see in Asia has been exposed to domesticated rice for a long time, and much of it represents escapes from domestication or where there's been gene flow from domesticated rice into those wild gene pools. In fact, much of the native rice area in Asia has actually been displaced by rice cultivation. So what we've got here is a reservoir of great diversity that hasn't been polluted by domestication, and it's allowing us to look at that. And so we're aiming to generate reference genome sequences for many of these populations, and I hope that ultimately taxonomists will come to provide names for them. They don't necessarily have species names at the moment, but they do represent novel relatives of rice. What the sequencing is allowing us for the first time to look at the gene pool of rice and do a phylogeny of all of the close relatives. And here's really our first, I suppose, coherent phylogeny of the genome species as we would see them at the moment, based on the whole chloroplast sequence. We have whole nuclear genomes that are being assembled for these. So this volume of data allows us to get phylogenies that are much more reliable than the ones in the past that have been based on individual genes and have usually been conflicting in the conclusions that we would draw. So what we would now see for rice, for example, is that there are two clades, an Asian clade and an African clade. Barthii here was, of course, domesticated to Glaberrima in Africa, and Rufi Pogon or something like it domesticated in Asia. There's more work to be done, I think, on really understanding the relationships in the Asian clade, but these two groups form a clade that is a sister group to the Australian wild population. So the Australian wild populations represent the first group within the genome that sits outside that domesticated pool. And then we see outside that again another clade that's found in South America and Africa that represents a greater genetic distance again. And this is all consistent with a southern hemisphere origin for the ICRISAT, and that we now have a much better understanding of structure and how we can search more widely for genetic diversity. These particular clades here represent really novel, untapped reservoirs for things like disease resistance in rice. We're beginning to see those exploited, but they really will represent great new opportunities for rice improvement. So some of these taxa look very much like Rufi Pogon, but they don't look like Rufi Pogon at the genome level. All like this is a perennial that looks a little bit like the annual meridionalis, but is in fact quite different at the genome level. One of the things that we can target looking at wild material is capturing genes that were lost in domestication quite specifically. And in this work here, we looked at where what we're calling the genome deserts are in rice. If you sequence lots of rice individuals and look at where the polymorphisms are, you do find areas like this 5 million base pairs here on chromosome 5, there's 1 million base pairs up here on chromosome 1, where there's essentially no variation in domesticated rice. Every individual has exactly the same sequence in these desert regions. But what we can find is in the wild material quite a lot of useful variation in those regions, and particularly we can find what are obviously disease resistance genes in in these deserts in the wild. And so we can be very deliberate about taking useful genes from wild material with this sort of insight as to where domestication has created a bottleneck. One of the areas that we're applying this into in rice, but looking more widely in the cereals, is in terms of starch structure. We know that we have very different starches in things like sorghum, rice and wheat, for example, and within them we're beginning to understand exactly what determines these differences, how we can make starch with any particular properties. And this is quite important because of the importance of starch in diets and the rising concern about diabetes and obesity and the contribution of high carbohydrate diets to that. And we believe that there's this potential to adjust the composition of these starches in ways that will be meaningful for human health at the global level, based upon understanding all of the genes in these pathways. So in rice, in the domesticated gene pool, we now believe that we understand, by looking at allelic diversity, by sequencing very large numbers of individuals, all the variation that explains the differences in the critical characteristics of the starch within the domesticated gene pool. What we're now doing is going out to the wild material and finding new alleles that aren't in the domesticated gene pool that allow us to think about even more radical adjustments in the chemistry of the starch and the properties of the starch within rice. And I think this is something that will extend beyond rice also to the other cereals. Also, I'd like to tell you something of the work we're doing with wheat. Wheat, of course, probably the other really important cereal. Here, I think the rate of genetic gain internationally is disappointing. There's a real focus in trying to work out how we can lift the rate of genetic gain in wheat. And one of the constraints is the need to make products like bread, that we really need to be conservative, and the need to make these end products really does put restrictions on what we can do in wheat breeding. So what we're trying to do is understand in much greater detail the molecular basis of wheat quality. And what we've been looking at is the transcriptomes of very large numbers of wheat varieties from around the world, and trying to look at which alleles are there that might tell us something about some of the reasons that we've been successful in developing high quality wheats in the past, and other cases where we haven't. And we have had a focus on understanding both chapati and bread quality, and that requires very different genetics, but we are starting to understand what they are. It would be great if we had wheats that you could actually make both chapatis and bread from at high quality, but at the moment there really are genetically fairly different wheats. What we've discovered is that there are some novel genes, rare alleles in the gene pool that really do seem to be quite critical in bread making. And this one here is a small protein that's expressed very highly through mid-seed development in wheat. It's one of the most differentially expressed genes we've been able to find in the wheat transcriptome. It differs 1,000-fold between genotypes, and those genotypes that express this protein all make high-quality bread. We found this allele in all of the prime hard or premium wheats that have been released commercially in Australia, and we don't find it in anything that's got poor quality. So some of these discoveries coming from sequencing are filling in gaps in our knowledge that came from the protein level. This is a very small protein, and all the people that have been studying wheat quality have been looking at big proteins. This would have run off the end of the gel, been in the die front in all the wheat chemistry work. And so it does show when you look at a different level. If you've been looking at the metabolite level, if you look at the proteome or the genome, you get completely new insights. And so there is evidence here that small proteins may play a really important role in wheat and may have been overlooked because the protein chemists weren't really looking for them. Another insight that's coming from genome sequencing is the work we've been doing here on hybrid rice with Advanta. We've been field trialling their progeny here in Hyderabad, and what we've been doing is sequencing the parents of their populations. And in things like maize, the wider the cross, the more heterosis, more or less. But in rice, people haven't been able to work out how to get heterotic groups, or how to make how to get that 20 or 25% yield gain that hybrid rice offers. And what we're seeing is that there seems to be an optimum. In these few cases here where we look at crossing something with things that are further away, about 250,000 SNPs seems to give us optimum heterosis. So the cross can be too close or too wide, and there seems to be an optimum. We're testing more and more populations, but this may be the reason that it's been hard to work it out, that it's actually quite quantitative. It seems to be it's about one polymorphism per gene across the genome is giving us maximum heterosis, but that you could have too little or too much. We've got both inbreeding and outbreeding depression, I suppose, in the classical genetic sense. So there's more work to be done on this, but this I think is a potentially useful insight into how we might be much more deliberate in getting those sorts of yield gains in things like rice. I'll just say, I'd like to say a little bit about some of our work on some other species, particularly the fruit crops. I think there's been a lot of work on genetic improvement in the temperate species that we're trying to extend to tropical species. The priorities for us are things like avocado, macadamia and mango, and we've just started a mango collaboration here in India with an effort to develop and exploit a mango genome, and we're doing the same sort of thing with macadamias and with avocados. And the sorts of things here we're trying to do is to develop, reinvent these species as smaller trees, much more higher in productivity, and to increase productivity now by as much as tenfold in terms of production per unit area. And we believe that's possible with intensive reinvention of these fruit crops. Comparative genomics has proved, I think, quite powerful in some of these systems, and we, with people at Washington State University and in Denmark, compared the cherry, the peach and the almond a few years ago at the whole genome level, and it seemed a naive experiment in some ways, but that actually allowed us to identify some functionality and the reasons for some of the differences between these species. So just aligning the sequences of genotypes of these species has proved very insightful. We've been able to identify the genetic basis of bitterness in almond, something that was really quite important, and understand much better the differences in ripening between the peach and the cherry, because we can see systematic differences in particular genes in the genome. And these small genomes are very easy to work with, and really whole genome sequencing is a really critical tool. We've applied this to looking at the wild relatives of the macadamia and discovered relationships we didn't realise before that are geographically based, and we've found that two small stature species are actually quite closely related to the main domesticated species, and this gives us an option to develop smaller stature trees by working with those other wild relatives. I'll just mention coffee. We've had a coffee consortium, and coffee is very important in that it is produced in developing countries in the main and sold in developed countries, so it shifts a lot of money in a desirable way in the world, and it is the highest value of our agricultural commodities at the global level. Sequencing the Robusta genome gave us some real insights. I think the thing that we took away, and this is the publication from Science last year, was that the caffeine, which is such a critical component of this product, evolved independently in coffee to tea and in chocolate. So we've got methyltransferases that are involved in biosynthesis of caffeine, but they're different methyltransferases, different branches of that family that have evolved this function in coffee, in tea and in chocolate. So here we have convergent evolution. This suggests that caffeines biologically may be much more important than we thought. It's led us to think about what it is that's so special about caffeine that plants would independently develop the ability to make it. And so we're gaining a lot, I think, of that sort of biology by doing this genome sequencing. We're trying now to broaden the gene pool for coffee, because coffee, Arabica coffee, of course, is a tetraploid, a recent origin, went through a significant genetic bottleneck. We're now looking at all of the species worldwide. The genus has expanded out now to be about 100 or more, and trying to understand those. We've got full-scale genome sequencing efforts in I think 17 of these species now that are going to allow us to really understand where we can go in the genus to bring new characteristics into coffee. And there really is some quite diverse material there. So we've got a fairly major consortium that's coordinating this effort. What we've found already is that we've got some surprises. This is an Australian species here, but this is Arabica coffee, and we've got Robusta here. What we know that Robusta is a parent of Arabica, but what we've identified in this is that that Robusta itself seems to have a hybrid origin, in that it's got part of its genome seems to have come from another genus. So we've got quite a number of breakthroughs in our understanding of the origin of coffee coming from sequencing the genomes. I'd just like to mention peanut because of where we are today. We're exploiting the emerging peanut genome. We've designed a system for enriching the genes in the genome by designing probes for all of the genes in the annotation of the peanut genome. And we're using that to look at this trait, which is important to us, to find genes that control blanchability, or the ability to convert this to that. That's a genetic trait that's highly heritable. We believe we can get at those genes fairly readily by sequencing the genes, the gene space of genotypes with and without the trait, and that works at a fairly advanced stage at the moment. So I'd also like to mention some of the work we're doing on energy crops, because I think they really remain important despite the low price of oil. I think that it won't be low forever, and we'll suddenly be wanting energy crops again. We're continuing our work on sugarcane and eucalypts because we see these as two of the big options in the world for energy crops. And we're really looking at cellulosic biofuels from sugarcane and from woody biomass, particularly in the form of eucalypts. The sugarcane sequencing effort is continuing on many fronts internationally. It's one of the hardest species imaginable to sequence. We've got have endless meetings and plans to do this. As you'd be aware, the genome is incredibly complex with two different species and some chromosomes being combinations of those, and with the different homology groups having different numbers of chromosomes. Really is very messy. So this is probably the last species that we're still coping with by doing back-by-back sequencing. So we're hoping this year to complete the sequencing of sufficient backs to have tiled the genome. But it may be the last genome that this is done in. I think the technology has continued to advance such that we've moved away from that approach in most species. But because of the complexity here, the very conservative approach of doing back-based sequencing is persisting. But I am optimistic we'll get there. I know when I talked last year, I said we're going to finish this year. This year I'm saying we're going to finish this year. I hope that's true, and I'm not saying it next year. The eucalypts are, in contrast, very easy. Very small genomes, 300 megabase, or smaller some of the ones we're looking at, but we've got 1,000 taxa, and we really don't know which ones we should be working with. And what we find is that it's hybrids between the species that give us all of the great biomass growth rates in marginal environments in so many places in the world. What we have confirmed by doing genome sequences now of many of the key species we think will be important for bioenergy is that we have widespread reticulate evolution going on in the eucalypts. So here we have 1,000 species, and if we look at the chloroplast of these, we find that the individuals in any one location all have the same chloroplast. So what we see is the species are interbreeding at a rate that allows the maternal genome to be shared locally, but not at such a rate that we see a breakdown of the morphological differences between the species. So that sort of dynamics I think we're finding in more and more wild plant systems. It says something about how these plants can evolve in such a way that there is this underlying reticulate evolution in the background, and that's partly what we're seeing, I think, in the coffee system as well. The sort of way that this will be useful to us is illustrated here with two species that the taxonomists have no difficulty in, and this slide doesn't really show the morphology. This one has small leaves, and this one has much bigger leaves. This is Henryi and this is Variegata. I can't find any difference between them at the DNA sequence level, and I think it's a single gene for size. The taxonomists find 10 differences, but they're the size of the leaves and the size of the flowers and the size of the fruit. So I think it's a gene that determines plant architecture or size. But now that we've got whole genome sequences, I believe we're going to be able to find maybe the gene for plant size, which could be actually a really important gene more widely in agriculture if we want to change the size of trees. So I think it's a good example of how genome sequencing can target what might be important discoveries that might, in this case, be applicable to the work we're doing in fruit trees, for example. I want to just finish by emphasising the fact that I see the future for the work that we're doing in this area is really information technology. It's incredibly data rich, working with plant genomes, and if we're thinking about the fact that the genotyping might be the genome sequence of every individual that we work with as plant breeders, for example, then we do have large amounts of data, and we have to learn how to deal with that, how to store it, how to manage it, and how to mine it in efficient ways. And I think that's where all of the real efforts that lie ahead of us are. I think we're getting a much better understanding of the relationships between plants at these broader levels. I think the phylogeny we now have for the flowering plants is quite different to the one we would have had 10 years ago, and much of that comes from our continuing understanding of evolutionary relationships that are coming from the much deeper sequencing we now have of plants across the whole of the plant kingdom. But that same sort of relationship that's getting much clearer, and it's really quite different, the view we would have of flowering plants than what we had 20 years ago. That's also translating down to the lower level within the groups that are related to our major crops. And those insights are really quite powerful. The one initiative that I think that really is bringing this together is the Dipseek initiative, and I think that that's one way in which we are going to internationally coordinate the capture of the sequences of all of the seed in our international seed collections. So the ambition here is certainly to sequence everything that might be held in a seed bank anywhere in the world and make all of that available in the public domain. And I think that's a very worthwhile objective, and I think that's one way in which we'll see a lot of this come together. I'd just like to finish by advertising a conference that we'll be hosting in Brisbane later in the year, Tropic 2015, where we'll be focusing on understanding the constraints to productivity in tropical agriculture. So I do hope you might consider coming and joining us for that meeting. I just finished by acknowledging and thanking the people in my research group. Thanks very much.
So many thanks for that tour de force. Maybe time for one or two quick questions?
Dave.
Sorry, you've expressed the need to keep on improving crops and expand the technology that we've got to a range of other crops, which I agree is crucially important. Do you see any breakthrough technologies on the horizon? You mentioned that it's difficult to predict what happens, but there are regular revolutions. Do you see anything that's on the horizon that could be coming up? And also, what should we be doing in terms of preparation for that? It's quite easy to get fixed on certain technologies and invest in that and get stuck with that, but it's to be able to remain flexible. Is there any suggestions around that?
Yeah, I think that's a really good point. I think the challenge we all face in this business, if you look back over that historical perspective I tried to give on the turn of the century, is that most of us stuck with technologies for too long and didn't move on to the next one. And I think that's the challenge we face ahead. I've got no doubt the breakthroughs are there. I can't tell you what they are, but I'm confident they'll happen. And the really important thing is that we don't keep doing what we're doing now, and we actually adapt that technology and apply it quickly. And that philosophy still probably doesn't get espoused enough, but we really have to be nimble with the technology, and it really will change. I think we are going to be doing that. I think the sort of efforts that we're seeing with Dipseek, for example, are readying us for that. We're not saying, no one's saying how we're going to sequence the genomes, but we're saying we are going to be able to get the sequence of everything. Let's plan for that. Let's think about how we're going to manage the relationships between the groups, the intellectual property issues and so forth, so that when the technology is invented, knowing that it will be invented very soon, we can do it as quickly as possible.
Okay, great. Thanks very much.